The school detonates before the strike even lands. Silver bodies flash in every direction, muscles firing in reflex arcs that have saved their lineage for millions of years. A single shadow knifes through the formation, and the water fractures into shards of movement. The prey do exactly what evolution trained them to do: scatter. And in scattering, they surrender the only protection they had. The predator does not chase the school. It waits for the moment the school dissolves. The executor of this calculated chaos is the great barracuda, a pelagic hunter built not for endurance, but for violent acceleration. Its body is a torpedo of muscle and bone, elongated and rigid, optimized for sudden bursts rather than sustained pursuit. The barracuda does not need to corner prey into caves or lure them into crevices. It weaponizes their own panic response. Schooling fish rely on cohesion. Their safety lies in synchronized motion, a strategy known as the dilution effect, where the probability of any one individual being targeted decreases within a dense group. The geometry of the school confuses predators, making it difficult to isolate a single body from the flashing mass. But that defense depends on unity. When cohesion breaks, each fish becomes a singular target. The barracuda understands this without conscious thought. It positions itself just outside the edge of the school, motionless, its reflective scales mirroring the environment. Those scales are not merely camouflage. They disrupt visual detection by fragmenting light across the body's surface, making it harder for prey to judge orientation and distance. The predator appears both near and far, a distortion in the water rather than a defined threat. The attack begins with speed that exceeds what most reef fish can generate. Powerful caudal fin strokes, driven by dense, fast-twitch muscle fibers, propel the barracuda forward in a rapid burst. But the strike is not aimed at a specific fish. It is aimed at the integrity of the school. The goal is disruption. As the barracuda slices through the mass, the lateral line systems of the prey are overwhelmed by sudden pressure changes. Individual fish initiate escape responses in random vectors, prioritizing immediate separation from perceived danger. The synchronized formation collapses into chaos. In that chaos, the barracuda recalibrates. Unlike predators that rely on prolonged pursuit, the barracuda executes a second phase, targeted interception. Its large eyes provide acute binocular vision, allowing it to lock onto isolated individuals in the aftermath of the scatter. Once a fish is separated from the group, its survival probability plummets. The barracuda accelerates again, jaws opening to reveal rows of dagger-like teeth designed to slice rather than crush. The prey's mistake was not slowness, it was isolation. The biomechanics of the bite reflect specialization. Barracuda teeth are laterally compressed and razor-sharp, capable of inflicting deep lacerations that sever muscle tissue and vital structures in a single snap. The predator often delivers a slashing bite, allowing the prey to weaken before a final capture. This method reduces the need for prolonged struggle and conserves energy. Yet the ecological twist reveals that the barracuda's dominance is dependent on the very behavior it exploits. If schooling fish maintained cohesion under attack, the barracuda's success rate would drop significantly. The predator thrives on panic, but panic is not universal. In some species, tight schooling persists even under intense predation pressure, forcing the barracuda to adapt its tactics. Control of chaos requires predictable prey responses. Furthermore, the barracuda's own survival hinges on precision. It lacks the venom of lionfish or the armored resilience of groupers. Its slender body, while ideal for acceleration, offers little protection against larger predators such as sharks. A miscalculated strike could place it within the jaws of a superior hunter. Thus, the barracuda often patrols near reefs or structures that provide quick retreat options. Its control over panic extends beyond immediate kills. By repeatedly triggering scattering events within certain reef zones, barracudas influence prey distribution patterns. Fish may avoid areas known for frequent high-speed attacks, altering grazing behavior and microhabitat use. The ripple effect of panic shapes the spatial ecology of the reef. The predator's reflective scales serve another function in chaos control. In bright tropical waters, Flashing light off its body can momentarily disorient prey. The sudden glint mimics the shimmer of multiple fish, creating visual confusion during the initial strike. Prey struggle to determine the exact location of the threat. That hesitation compounds the fragmentation of the school. The reversal emerges when observing juvenile barracudas. Young individuals often form loose aggregations, schooling themselves. In early life stages, they rely on the same cohesion strategies as their prey. Panic is something they must avoid, not create. Only as they mature and their speed and jaw strength increase do they transition into solitary chaos engineers. The predator once depended on unity before learning to dismantle it. The barracuda's nervous system is tuned for rapid processing. High visual acuity and fast neural conduction allow precise timing during split-second events. In chaotic environments, reaction speed determines success. The predator must interpret dozens of moving targets and select one within milliseconds. As the scattered fish attempt to reform their school, the barracuda circles, waiting for the next vulnerable moment. It does not pursue the regrouped mass indefinitely. It waits for weakness. Another burst, another rupture, another isolated body caught mid-turn. The water gradually settles, but the geometry of trust within the school has been shaken. Individuals that fled too far rejoin cautiously. The memory of sudden disruption lingers in their collective movement. Control through panic is subtle. The barracuda does not herd prey into corners or lure them with deception. It detonates order. It fractures the defensive architecture of the group and capitalizes on the fragments. In ecosystems where barracudas are abundant, schooling fish exhibit heightened vigilance, maintaining tighter formations and adjusting swimming depth. The predator's influence extends into behavior even when it is absent. The mere possibility of sudden chaos alters how prey move through space. The barracuda's dominance lies in timing the fracture. It strikes not to overpower the entire group, but to shatter it. Once unity dissolves, the outcome is determined. The school's instinct was to scatter, and that instinct has preserved countless generations. Yet in the presence of this torpedo-shaped predator, scattering becomes surrender. The barracuda does not need to corner its prey. It controls the moment they lose coordination. And in that moment of chaos, it rains. The light begins to pulse before the fish understands that it is already late. Bands of electric contrast ripple across the sand, dark to pale, pale to dark, in strobing waves that do not illuminate the seabed so much as fracture it. The small wrasse hovering above the substrate adjusts its pectoral fins, recalibrating balance in the mild current, and then something impossible happens. The ground itself seems to move. Stripes cascade forward in hypnotic succession, expanding and collapsing with surgical rhythm. The wrasse does not flee. It stalls. And in that infinitesimal stall, the strike is launched. The architect of this moment is the common cuttlefish, a cephalopod whose mastery lies not in brute force, but in the manipulation of neural timing. Where other predators overwhelm through speed or stealth, the cuttlefish intervenes directly in the prey's sensory processing window. It does not chase first. It edits perception. The cuttlefish's skin is a living display panel layered with chromatophores, iridophores, and leucophores, each controlled by motor neurons firing with millisecond precision. Unlike pigment changes governed by hormones in many animals, these color shifts are immediate because they are wired directly to the nervous system. When the cuttlefish deploys a behavior known as the passing cloud or the mesmerizing display, it sends coordinated neural signals across its mantle that create waves of high contrast patterns traveling along its body. These waves do not simply alter appearance, they exploit the prey's visual circuitry. Small fish rely heavily on motion detection to trigger escape responses. Their retinas are tuned to sudden changes in contrast, and their midbrain structures process looming stimuli as imminent threat. The cuttlefish's pulsing patterns generate conflicting signals. The stripes mimic forward motion while the body remains nearly stationary. To the wrasse, the substrate seems unstable, as though the entire visual field is shifting. Neural resources that should be allocated to spatial orientation are diverted to resolving contradictory motion cues. Reaction time elongates by fractions of a second, yet those fractions are decisive. At the core of this strategy lies an understanding encoded by evolution. Prey survival depends on rapid, stereotyped responses. When startled, a fish initiates a C-start escape reflex, bending its body into a tight arc before launching away. This reflex is governed by giant mouth neurons that fire almost instantaneously upon detecting threat. The cuttlefish's display interferes not with muscle strength but with the trigger threshold. By overwhelming visual input with rhythmic patterning, it delays the sensory confirmation needed to activate that reflex. The fish hesitates, recalculates, and in that recalculation, the cuttlefish advances. The strike itself is a biomechanical marvel. Two elongated tentacles, normally concealed within specialized pockets, are propelled forward by a rapid muscular contraction that creates hydrostatic pressure. The tentacles extend faster than the fish can compensate, the adhesive clubs at their tips locking onto the prey's body. The control is immediate and absolute. The fish does not outswim the predator because it never receives the signal to flee in time. Yet the cuttlefish's control is not limited to prey. It also manipulates rivals and potential mates through dynamic skin signaling. Males competing for females display vivid patterns that communicate strength and readiness. In some cases, smaller males adopt split body signaling, presenting a dominant pattern toward a female while simultaneously displaying a female-like pattern toward a rival male. This bilateral deception reduces aggression from competitors while maintaining reproductive opportunity. Neural control over skin becomes social leverage. The sophistication of this system is anchored in a brain that, relative to body size, rivals many vertebrates in complexity. The cuttlefish optic lobes are highly developed, processing vast amounts of visual data in real time. This visual acuity supports not only camouflage, but also the precision of disruptive displays. It must analyze the prey's orientation, distance, and angle of view to deploy the most effective pattern. Control here is contingent upon accurate sensory modeling. The ecological reversal emerges when one considers that the cuttlefish is itself vulnerable to numerous predators, including dolphins, larger fish, and even seals. Its soft body offers little protection, and its internal shell, the cuttlebone, provides buoyancy regulation rather than armor. Neural disruption evolved not as excess, but as necessity. By ending encounters swiftly through reaction manipulation, the cuttlefish minimizes exposure time in open water. Its mesmerizing display is not flamboyance. It is risk mitigation. Even more intriguing is that cuttlefish can modulate these displays depending on environmental light conditions. In low visibility waters, contrast patterns are amplified, whereas in bright shallow zones, subtler gradients suffice. This adaptability suggests continuous feedback between environmental luminance and neural output. The predator calibrates its own signal strength to maximize cognitive overload in prey. The passing cloud display, often interpreted as camouflage, doubles as a psychological tool. While it can obscure outline against dappled light, it also creates the illusion of motion across a stationary body. Prey accustomed to scanning for moving silhouettes struggle to pass a predator that appears simultaneously still and in motion. The cuttlefish occupies the blind spot between detection and recognition. And yet, the final reversal challenges our assumption of domination. The cuttlefish's mastery over timing is balanced by its own temporal fragility. Most species live little more than a year, investing heavily in reproduction before senescence. After spawning, adults often decline rapidly, their neural vigor fading as metabolic priorities shift. The animal that controlled milliseconds of reaction loses control over the arc of its own lifespan. Its brilliance is compressed into a fleeting window. Within that window, however, it rewrites the rules of predation. It does not rely solely on invisibility, though it can vanish into coral rubble with breathtaking fidelity. It does not depend purely on speed, though its tentacular strike is among the fastest in the invertebrate world. Instead, it intervenes at the level of cognition. It edits the prey's decision-making process, inserting confusion where certainty should exist. As the wrasse drifts above the sand, its sensory world remains coherent until the first ripple of contrast arrives. The seabed flickers, the pattern intensifies, and neural circuits designed for survival are momentarily overwhelmed. That moment is enough. The tentacles launch, adhesive pads secure flesh, and the wrasse's body is drawn toward the beak concealed within the mantle. The cuttlefish releases no roar, no visible sign of triumph. Its skin settles into muted tones. Its mantle resumes gentle undulation. The ocean appears unchanged. Yet in the silent gap between stimulus and response, a predator has seized control not of muscle, but of time itself. By manipulating the interval in which a decision is made, the cuttlefish dictates outcome. The prey moves, but it moves too late. Reaction becomes hesitation, hesitation becomes capture, and the rhythm of survival bends to the precise, luminous choreography of neural disruption. The water never explodes. It stretches. A sardine breaks from the school, sensing pressure in the current, and launches into open blue. Behind it, a shape does not sprint. It settles into rhythm. There is no violent acceleration, no theatrical burst of speed, only a tightening arc that begins to close with mathematical patience. The sardine darts left, then right, then upward, muscles flooding with lactate as anaerobic pathways ignite. The predator adjusts, not with panic, but with persistence. The distance between them does not collapse instantly. It erodes. The architect of this erosion is the Atlantic bluefin tuna, a pelagic predator whose dominance lies not in ambush or deception, but in physiological supremacy. It does not manipulate prey behavior through illusion or terrain. It manipulates time. It decides not where the encounter ends, but when the prey's body gives out. Tuna are engineered for endurance at a scale few fish can match. Their bodies are rigid, streamlined torpedoes built for sustained high-velocity swimming, with crescent-shaped caudal fins that reduce drag while maximizing thrust efficiency. But the true advantage lies beneath the skin. Unlike most fish, which are ectothermic and whose body temperature mirrors surrounding water, bluefin tuna possess regional endothermy. Through a countercurrent heat exchange system known as the rete mirabile, they conserve metabolic heat generated by red muscle activity, maintaining core temperatures significantly above ambient water. This thermal advantage translates into power. Warmer muscle fibers contract more efficiently, generating greater force and faster reaction times. While sardines and mackerel must operate at the mercy of surrounding temperature, the tuna carries its own engine. Its cardiovascular system supports high oxygen delivery through large gill surface area and a robust heart, enabling sustained aerobic metabolism. The sardine's burst is anaerobic and brief. The tuna's pursuit is aerobic and prolonged. The chase is not reckless. The tuna rarely engages in a single blind sprint. It aligns itself behind fleeing prey, matching speed just below maximum output. This strategy prevents premature fatigue in the predator while steadily forcing the prey into metabolic crisis. Each dart the sardine makes consumes glycogen reserves. Each evasive maneuver accumulates lactic acid within muscle tissue. The tuna's red muscle fibers, rich in myoglobin, continue receiving oxygenated blood, clearing metabolic byproducts efficiently. Exhaustion is not sudden collapse. It is incremental failure. The sardine's turning radius widens slightly. Reaction time slows by fractions of a second. The tuna notices. Its large eyes, adapted for clear pelagic vision, track minute changes in tail beat frequency. When the prey's cadence falters, the tuna accelerates, narrowing the final gap in a decisive surge. The kill is swift. The control was gradual. This form of dominance is intimately tied to hydrodynamics. Tuna travel in open water where there are no choke points, no caves, no coral to exploit. The battlefield is endless blue. In such terrain, territorial ambush is impossible. Instead, selection has favored energy efficiency and stamina. The tuna's pectoral fins retract into grooves at high speed, minimizing drag. Its rigid body reduces lateral flex that would waste propulsion energy. Even its scales are small and embedded, smoothing flow. But the ecological twist complicates the image of an unstoppable endurance machine. Tuna are obligate ram ventilators, meaning they must keep swimming to force water across their gills. Stopping is not an option. The same physiology that grants them stamina also binds them to perpetual motion. A tuna cannot hover in place like a grouper or rest within a crevice like a moray eel. If forced into constrained environments or entangled in nets, it cannot simply cease movement without suffocating. Its supremacy in open water is vulnerability and confinement. Furthermore, the tuna's control over exhaustion shapes pelagic ecosystems. By targeting weaker or slower individuals within prey schools, tuna exert selective pressure that strengthens prey populations over time. Only the fastest sardines survive repeated encounters, refining the evolutionary arms race. The tuna does not merely consume prey, it sculpts their physiology. Long chases also influence schooling dynamics. Sardine schools often tighten formation when tuna are present, attempting to confuse the predator. But tight schooling reduces oxygen availability within the group as individuals compete for water flow across gills. In attempting to conserve energy collectively, they may inadvertently increase stress. The tuna, circling patiently, waits for the moment when cohesion fractures under metabolic strain. The predator's own endurance is not infinite. Sustained pursuit demands enormous caloric intake. Bluefin tuna undertake long migrations across ocean basins, following prey availability and temperature gradients. Their muscle mass and high metabolism require constant feeding. In seasons when prey density declines, even the endurance master must travel vast distances to sustain itself. Control over exhaustion applies to prey, but the tuna remains bound to environmental productivity cycles. There is also a social dimension to this strategy. While often depicted as solitary hunters, tuna can coordinate loosely within schools of their own species. When multiple individuals pursue the same prey aggregation, they can drive schools upward toward the surface, reducing vertical escape options. In these moments, exhaustion is compounded by spatial restriction, as prey are compressed between predators below and open air above. The final reversal lies in scale. The tuna's dominance over individual sardines does not grant control over the ocean's vastness. Climate shifts that alter water temperature can affect the efficiency of their heat exchange systems. Overfishing can deplete their numbers despite their speed and stamina. The animal that dictates when prey fail cannot dictate when its own population declines. As the sardine's body begins to wobble under lactic acid burden, its escape arcs flatten. The tuna senses the weakness and closes the final meters in a controlled acceleration. There is no chaos, no dramatic spectacle. The ocean remains blue and vast. The decisive factor was not terrain or illusion, it was endurance. In this open realm, control is measured in seconds shaved from stamina, in oxygen delivered more efficiently, in muscles that resist fatigue longer than those of any prey. The tuna does not ambush or trap. It waits for the inevitable moment when biology yields. The sardine's failure was not a mistake of direction, it was a failure of time. The tuna governed that time from the first turn of its tail. In the endless blue where there are no walls to close and no shadows to hide within, the only boundary is physiological limit. And the tuna stands at that boundary, deciding precisely when it will be crossed. The reef corridor narrows to a single opening no wider than a doorway, and the current accelerates as it is forced through stone. A cloud of glass minnows drifts with the flow, trusting the geometry of the passage to shield them from open water hunters. The entrance ahead is shadowed but calm, a pocket of reduced turbulence that promises relief from the push of tide. The minnows angle toward it in unison. They do not see the boundary. They cross it. The shadow inhales. The predator waiting at that threshold is the Goliath grouper, a reef titan that exerts dominance not through pursuit, but through territorial architecture. Its body, thick and muscular, seems ill-suited for speed, yet it does not need velocity. It positions itself precisely where movement must compress, at cave mouths, overhangs, channel bottlenecks and coral arches, where prey have no choice but to pass. Control here is not over individual fish, but over the pathways that define their world. Groupers are masters of ambush. Their laterally compressed bodies allow them to wedge into tight entrances, blending with mottled rock through cryptic coloration. From a distance, the fish appears to be another slab of coral shadow. Only the slight rhythmic opening of its mouth betrays respiration. That respiration, powered by buccal pumping, keeps water flowing across gills while the body remains still. Stillness is essential. Prey navigating reef channels rely on visual scanning for sudden motion. The grouper denies them that cue. The first phase of choke point control is site selection. Reef systems are not uniform landscapes. They are networks of corridors shaped by hydrodynamics and coral growth. Currents funnel plankton and small fish through predictable routes, especially during tidal shifts. Groupers learn these routes. Through spatial memory and repeated use of territory, they identify locations where prey density peaks. By occupying the narrowest section, they reduce the number of escape vectors to 2, forward or backward. When prey are carried by current, backward retreat is costly. Forward becomes default. The strike itself is explosive. Like other ambush predators, groupers employ rapid suction feeding, expanding their buccal cavity to create a vacuum that pulls water and prey inward. But what distinguishes them is timing relative to terrain. They do not strike in open water where prey can scatter. They strike at the exact moment prey enter the choke point, where lateral escape is limited by stone walls. The physical space amplifies suction efficiency. The biomechanics are formidable. Large groupers can generate significant negative pressure within their oral cavity, aided by strong cranial musculature and flexible ligaments that allow rapid jaw protrusion. The mouth opens wide enough to engulf multiple small fish simultaneously. Because the prey are compressed by the narrow passage, the grouper's single suction event can capture several individuals at once. Yet the ecological twist complicates this image of solitary dominance. Groupers are not always lone strategists. In some reef systems, they participate in cooperative hunting with moray eels. The grouper patrols open water edges while the eel invades crevices. If prey escape the eel by darting outward, the grouper intercepts them. This partnership effectively seals both horizontal and vertical escape routes. The choke point expands beyond a single cave entrance into a multi-species blockade. Even without cooperation, the grouper's presence alters prey behavior. Small reef fish often avoid certain overhangs and caves known to harbor large predators. This avoidance shapes grazing patterns and micro-habitat use. Areas near active grouper dens may experience different algal growth due to reduced herbivore activity. Thus, the grouper's control over movement influences ecological structure beyond immediate predation events. The reversal lies in the grouper's own vulnerability. Despite their size, groupers are not apex predators in all contexts. Large sharks and human fishers target them, and their tendency to aggregate for spawning makes them especially susceptible to overfishing. Their reliance on fixed territories and predictable choke points becomes liability when faced with intelligent predators. What serves as a trap for minnows becomes a predictable ambush site for nets and spears. Groupers are protogynous hermaphrodites, meaning many species begin life as females and transition to males later. This reproductive strategy ensures that dominant individuals can control breeding opportunities within a territory. Social control within their own species mirrors spatial control over prey. Large males often occupy prime habitat, defending caves and ledges that double as feeding choke points and spawning sites. The control of space thus extends into control of reproduction. Their sensory systems are finely tuned to the dim light of reef cavities. Large eyes gather available photons, and their lateral line detects subtle water vibrations. Prey entering a cave mouth may not see the grouper, but the grouper senses their approach through pressure changes and microcurrents. It calculates timing based on hydrodynamic cues as much as visual confirmation. The reef architecture itself becomes an extension of the grouper's body. By positioning in narrow passages, the fish leverages stone as part of its hunting apparatus. Coral walls replace lateral fins in blocking escape. The cave ceiling restricts upward flight. The grouper's mass occupies the remaining gap. Prey that seek refuge from open water predators inadvertently funnel themselves into a controlled environment. As the glass minnows are drawn inward by suction, the event is nearly silent. A swirl of scales, a flash of white gill arches, and then absence. The cave entrance returns to shadow. Outside, the current continues, carrying more prey along the same route. The grouper does not roam widely. It waits. Patience is a metabolic strategy. Larger body size confers greater energy reserves, allowing extended periods between successful strikes. By conserving energy and relying on choke points, the grouper minimizes unnecessary movement. In ecosystems where groupers are abundant, their choke point strategy contributes to top-down regulation. By removing significant numbers of mid-level predators and herbivores, they influence trophic cascades that shape reef resilience. The presence or absence of large groupers can determine whether certain fish populations proliferate unchecked or remain balanced. The final reversal emerges when observing how prey adapt. Some fish species alter schooling depth or adjust timing of movement to avoid peak grouper activity. They learn the rhythm of the choke point. Yet complete avoidance is impossible in complex reef systems where passages are limited. The architecture that provides shelter from one threat inevitably exposes them to another. The cave entrance remains the focal point. It is neither fully open nor fully closed, a liminal space where safety and danger intersect. The grouper occupies that threshold, transforming geography into weapon. It does not chase prey across expanses of coral. It governs the gateways through which prey must pass. In doing so, it dictates the flow of life through stone corridors. The minnows believed they were entering refuge from current. Instead, they entered a domain already claimed. The choke point is not merely a location. It is a decision trap embedded in architecture, where movement is funneled into inevitability, and the boundary between freedom and capture is no wider than a mouth hidden in shadow. A reef falls silent, without a single predator striking. Fish approach the limestone slope and turn away, not because something chased them, but because the ground itself has grown teeth. Across the substrate spreads a dense, bristling carpet of spines, each one angled outward like a drawn blade. What was once open grazing territory has hardened into a field of deterrence. Movement slows. Roots shift. Entire feeding patterns dissolve. No jaws snapped shut. No fins accelerated. The boundary was established by stillness. At the center of this transformation is the long-spined sea urchin, a creature without speed, without venomous pursuit, without the mobility of most reef architects. It does not ambush prey. It does not inflate or chase. It reorganizes space itself. Through sheer density and structural defense, it converts reef surfaces into exclusion zones, controlling where others can settle, feed, and survive. The body of the sea urchin is armored by a rigid endoskeleton known as a test, composed of interlocking calcareous plates that form a near-spherical shell. From this shell extend spines that can reach lengths many times the diameter of the body. In species like the long-spined urchin, these structures create a moving thicket across the reef floor. Each spine is articulated at its base, capable of subtle motion that responds to water movement and threat. The animal does not strike outward. It forms a perimeter. Predatory fish that might consume softer invertebrates hesitate before a dense aggregation of urchins. The cost of contact is puncture, sometimes accompanied by mild venom delivered through spine surfaces in certain species. While not universally lethal, these injuries are painful and discouraging. Thus, a cluster of urchins becomes more than a collection of individuals. It becomes a collective barrier. Yet the deeper control is ecological rather than defensive. Sea urchins are voracious grazers of algae. Using a complex jaw apparatus called Aristotle's lantern, they scrape algae and biofilm from rock and coral surfaces. In balanced densities, this grazing prevents algal overgrowth that could smother coral polyps. However, when urchin populations surge, their feeding can strip reef surfaces nearly bare, removing not only algae, but also delicate encrusting organisms that stabilize coral skeletons. In such scenarios, the reef transforms into what ecologists term an urchin barren. These zones, common in temperate kelp forests and certain tropical reefs, are characterized by reduced structural complexity and diminished biodiversity. Coral recruitment declines. Small invertebrates lose attachment sites. Fish species dependent on branching coral for shelter migrate elsewhere. The urchins did not chase them away. They removed the architecture that sustained them. The narrative reversal complicates the idea of urchins as mere destroyers. In many reef systems, urchin populations are regulated by predators such as triggerfish, wrasses, and lobsters. When these predators decline due to overfishing or habitat loss, urchin numbers can expand unchecked. The barren landscape becomes not solely the urchin's creation, but a symptom of trophic imbalance. The spined field is both agent and indicator. Control arises from absence higher up the food web. In the Caribbean, a massive die-off of long-spined sea urchins in the early 1980s led to dramatic algal overgrowth as grazing pressure collapsed. Coral reefs that once balanced coral and algae shifted toward dominance by macroalgae. In this case, the disappearance of the urchin reshaped the ecosystem just as profoundly as its overabundance might have. Control operates in both presence and absence. The spines themselves function as more than deterrence. They create microhabitats within their forest-like arrangement. Small fish and invertebrates sometimes shelter among the spines, exploiting the barrier against larger predators. Thus, the urchin simultaneously excludes some species while protecting others. Its body becomes habitat, a living structure embedded within the reef matrix. Metabolically, sea urchins are efficient converters of primary productivity. By consuming algae and converting it into calcified structure, they integrate plant biomass into skeletal form that persists even after death. Empty tests contribute to sediment composition, influencing reef substrate characteristics over time. The impact extends beyond immediate grazing into geological contribution. The ecological twist sharpens when predator control returns. In regions where predatory fish populations recover, urchin densities often decline to balanced levels, allowing coral and algae to coexist. The once dominant no-go zone dissolves. The spined carpet thins and pathways reopen. The urchin's control is powerful but contingent. It thrives within specific predator-prey dynamics and collapses outside them. Even reproduction reflects ecosystem-scale influence. Many sea urchins release gametes into the water column during synchronized spawning events, often triggered by lunar cycles and temperature shifts. Massive numbers of larvae drift before settling, ensuring that population surges can occur rapidly under favorable conditions. When predators are scarce and algal resources abundant, recruitment may outpace mortality, reinforcing the transformation of reef terrain. Unlike the puffer that intimidates through spectacle or the stonefish that dissolves into substrate, the sea urchin imposes control through cumulative presence. One urchin is an obstacle. Hundreds reshape landscape. Thousands redefine community structure. As a reef fish approaches a slope densely populated with long-spined urchins, it must calculate risk and reward. Grazing algae between spines risks injury. Nesting among spines offers shelter, but restricts movement. Predators may avoid the area, but so might potential mates or competitors. The reef has been partitioned not by speed or aggression, but by geometry and grazing pressure. The final image is not of attack, but of territory converted into deterrence by abundance. The reef floor bristles under the weight of countless articulated spines, each one a slender assertion of space. In ecosystems shaped by subtle balances, the sea urchin demonstrates that control can emerge from density rather than dominance, from feeding rather than fighting. By scraping away the growth that builds coral complexity, or by preventing algae from overwhelming fragile structures, it directs the trajectory of entire reef systems. No chase unfolds. No venom is injected into prey. Yet corridors close, habitats shift, and species relocate. The urchin's dominion is written into the substrate itself. Where its spines cluster, movement narrows. Where its grazing intensifies, architecture simplifies. In the silent language of reefs, control does not always roar. Sometimes it bristles. The water fractures before the seal understands it has already been selected. A dark shape rolls beneath the ice edge, not striking, not accelerating, simply existing in a position that closes one escape corridor while leaving another temptingly open. A second shadow appears to the left, then a third behind. The seal's heart rate spikes. Lactate begins to accumulate in its muscle tissue as it prepares for anaerobic sprinting, but the ocean around it has already shifted from habitat to architecture. The hunters are not converging, they are arranging. The decision has been made long before the first burst of speed. The architects of this inevitability are the orca, apex predators, whose dominance does not rely on raw pursuit, but on coordinated manipulation of space itself. Each individual is a powerful swimmer capable of speeds exceeding 30 knots, but speed is rarely the decisive variable. Instead, orcas employ social intelligence, acoustic precision, and hydrodynamic positioning to shape the movements of entire groups of prey. Their control begins not with teeth, but with geometry. The pod operates as a distributed decision-making network. Vocalizations, many of them specific to familial groups known as matrilines, carry detailed information across the water column. These dialects are culturally transmitted rather than genetically encoded, meaning hunting strategies are learned, refined, and inherited socially. When targeting schooling fish, the pod does not scatter the shoal through chaos. It compresses it. Individuals flank the perimeter, releasing bursts of bubbles that form visual barriers, exploiting the prey's innate avoidance of contrast. The fish respond predictably, tightening their formation to reduce individual predation risk, a behavior known as the confusion effect. The orcas exploit this defensive reflex by herding the fish into a dense sphere, sometimes called a bait ball, where oxygen consumption increases and escape options collapse. The prey's survival strategy becomes its vulnerability. When hunting marine mammals, the control deepens. In subpolar regions, pods have been documented generating coordinated waves to wash seals from ice flows. Several orcas are lined side by side, accelerating in synchrony to produce a surge that destabilizes the platform. This is hydrodynamic engineering. The energy expenditure is shared across individuals, reducing metabolic cost per hunter, while the seal's refuge is transformed into a liability. The seal is not chased into the water, it is removed from its foundation. Control here is environmental manipulation on a massive scale. Yet the most chilling aspect of orca dominance is their ability to isolate. In large baleen whale migrations, pods may target a calf rather than confront the full defensive capacity of the adults. The pod divides into subgroups, some distracting the mother, while others maneuver to separate the young whale from its protective circle. This is not random aggression. It is strategic partitioning of social bonds. By exploiting the maternal instinct to pursue attackers, orcas create spatial gaps that widen into lethal distance. The calf's fate is sealed not by superior strength alone, but by calculated disruption of cohesion. This behavior is underpinned by neurological sophistication. Orcas possess some of the largest brains among marine mammals, with extensive cortical folding that increases surface area for neural processing. Their limbic system, associated with emotion and social bonding, is highly developed, supporting complex cooperative interactions. They do not merely hunt together, they anticipate each other's movements. Cooperative hunting requires predictive modeling, an ability to infer how a partner will act in the next moment. This cognitive foresight allows the pod to choreograph encirclement without collision, to adjust speed and angle dynamically as prey attempts escape. Even their echolocation contributes to control. By emitting clicks and interpreting returning echoes, orcas construct acoustic images of prey distribution and terrain. This sonar does not just locate individuals, it assesses density, movement, and orientation. The pod can determine which fish are injured or which seal is closest to the ice edge. Decision-making occurs before visual confirmation, granting the hunters informational superiority. In darkness or turbid water, where vision fails, acoustic mapping preserves dominance. But the narrative reversal emerges when examining prey responses. Humpback whales have been observed intervening in orca hunts, sometimes placing themselves between pod and victim. At first glance, this appears to challenge orca supremacy. Yet even here, control persists. Orcas adjust strategy, sometimes abandoning the target or switching to different prey classes altogether. Their ecological dominance is not defined by winning every encounter, but by adaptability across contexts. Some pods specialize exclusively in fish, others in marine mammals, and these specializations are culturally maintained. This dietary divergence reduces direct competition between pods and enhances resource partitioning within the species. Control extends beyond immediate prey to the structuring of the ecosystem itself. In regions where orca predation on sea otters has increased, cascading effects have transformed kelp forest dynamics. Sea otters regulate sea urchin populations, and without them, urchins overgraze kelp beds, leading to habitat collapse. Thus, the orca's decision to shift diet reverberates through trophic levels, reshaping entire coastal systems. The control of a single prey species influences carbon sequestration, fish nursery habitats, and shoreline stability. Dominance here operates at planetary scale. Yet the orca is not invulnerable. Its reliance on cooperative hunting demands social stability. Disruption of matrilines through human activity, such as ship noise or prey depletion, can fracture knowledge transmission. Without elders to guide hunting tactics, younger individuals may struggle to master complex strategies. The pod's greatest strength, cultural continuity, becomes a potential weakness under environmental stress. Their control depends on memory. Even within the hunt, restraint often precedes violence. Orcas have been observed stunning fish with tail slaps rather than immediately consuming them, effectively immobilizing multiple prey before feeding. This reduces the risk of escape and allows efficient energy allocation. The pod determines the tempo of the encounter. They accelerate when needed, decelerate when prey is exhausted, and occasionally appear to play with targets, though this behavior likely serves skill reinforcement rather than cruelty. The outcome is predetermined through attrition and coordination rather than chaotic struggle. Isolation remains their signature tactic. When targeting a pod of dolphins, orcas may force the group toward shallow waters, using acoustic harassment and high-speed chases to compress the formation. Dolphins rely on synchronized swimming and echolocation to maintain cohesion, but sustained pursuit depletes oxygen reserves. Orcas rotate lead positions, conserving stamina, while the prey cannot. The difference in respiratory strategy becomes decisive. Orcas, larger and with greater oxygen stores, can sustain longer dives and bursts. They choose when the encounter ends. The final reversal lies in perception. The term killer whale evokes singular ferocity, yet their dominance is profoundly communal. No individual orca controls the situation alone. Authority emerges from cooperation. The pod functions as a single adaptive organism, each member contributing sensory input, muscle power, and learned behavior. Their teeth deliver the final act, but the hunt's success was sealed by social intelligence long before contact. And so the seal that sensed danger too late was not defeated by speed. It was defeated by coordination. The fish compressed into a sphere did not succumb to chaos. They succumbed to engineered order. The calf separated from its mother was not simply overpowered. It was strategically isolated. Orcas do not dominate through impulse. They dominate through design. They decide the outcome before the first acceleration, shaping currents, herds, and destinies until resistance is reduced to inevitability. The reef tightens without moving. A cloud of juvenile damselfish hovers above a coral outcrop, their silver flanks flickering in the filtered sunlight, when the water behind them darkens, not with speed, but with structure. Striped spines unfurl like a closing gate. Broad pectoral fins spread into a living wall, each ray extended to impossible width, each membrane glowing with warning bands of white and rust. The fish do not scatter outward. They compress inward. The reef, once an open labyrinth of escape routes, has just become a corridor with only one direction left. The architect of this narrowing geometry is the red lionfish, a predator that governs through spatial domination rather than pursuit. Unlike fast pelagic hunters that overwhelm prey with acceleration, the lionfish rarely needs to sprint. It advances slowly, deliberately, fanning its immense pectoral fins outward until it occupies more volume than its body alone would suggest. Those fins are not ornamental, they are partitions. The spines along its dorsal ridge are venomous, each connected to glandular tissue capable of delivering painful neurotoxic injections. To the damselfish, the expanding silhouette is not merely larger, it is hazardous. Prey fish operate within a constant negotiation between flight distance and shelter proximity. On coral reefs, safety lies in complexity. Crevices and branching corals offer micro-refuges where small fish can dart beyond the reach of predators. The lionfish disrupts this equation by inserting itself between prey and refuge. As it spreads its fins, it blocks lateral escape, forcing fish toward a narrowing wedge of open water. Every slow pulse of its pectoral membranes pushes a subtle pressure wave forward, encouraging prey to retreat exactly where the predator intends. The venomous spines amplify the effect. Lionfish do not need to strike with these spines during a hunt. Their mere presence enforces distance. Prey species have evolved to avoid sharp silhouettes that suggest injury risk. The extended spines create a perimeter that prey instinctively avoid crossing. The lionfish thus establishes a no-entry zone defined not by speed, but by threat. Movement options collapse because the cost of brushing against a spine is high. As the damselfish school compresses, individuals begin to separate from the safety of coral branches. The lionfish adjusts its orientation, angling its body so that its wide fins form a semicircular barrier. Its head lowers slightly, jaws poised. Unlike suction specialists such as monkfish, lionfish employ rapid forward lunges powered by caudal fin thrust. But that lunge is short and decisive, executed only when prey are confined within a tight arc. The hunt is over before it begins because the geometry has already been solved. This strategy is rooted in biomechanics and ecology. Lionfish are not built for endurance chases. Their ornate fins create drag, reducing sustained swimming efficiency. Instead of evolving streamlined speed, they evolved surface area. That surface area becomes a movable fence. By expanding their physical profile, lionfish convert three-dimensional reef space into a two-dimensional corridor. The ecological consequences of this spatial control are profound. In regions where lionfish populations have expanded beyond their native Indo-Pacific range into the Western Atlantic and Caribbean, they have dramatically reduced juvenile reef fish abundance. Native prey species, unfamiliar with lionfish hunting style, fail to recognize the threat of the spreading fins until it is too late. The predator's control over movement options translates into measurable declines in biodiversity. Invasive populations can consume large numbers of juvenile fish daily, altering recruitment rates and shifting reef community composition. Yet the narrative reversal complicates this apparent dominance. In their native habitats, lionfish coexist with predators that recognize and sometimes consume them, including certain large groupers and sharks. Their venom deters many attackers, but does not render them invincible. Moreover, their elaborate fins make them conspicuous. In ecosystems where prey have co-evolved with lionfish, escape responses are sharper and more immediate. Spatial control is not absolute, it is context-dependent. The lionfish's sensory system further refines its strategy. It relies heavily on vision, with eyes positioned to track small prey movements within cluttered reef environments. Unlike predators that strike at random, lionfish often stalk slowly, adjusting fin angle based on prey orientation. Some observations suggest cooperative hunting behaviors, where two lionfish may align to herd prey into a confined pocket. Although not as socially complex as dolphins or orcas, this occasional alignment demonstrates flexibility in spatial manipulation. Venom, though rarely used offensively in hunting, reinforces control indirectly. The venom apparatus consists of grooves along the dorsal, pelvic, and anal spines, through which toxin is delivered upon contact. The toxin causes intense pain and can disrupt neuromuscular transmission in attackers. Predators that have experienced such envenomation are unlikely to repeat the mistake. Thus, the lionfish carries a perimeter of remembered danger. Even without deploying venom during a hunt, the threat of it shapes the behavior of other species. As the damselfish group is funneled toward a rocky overhang, the lionfish's fins create a narrowing triangle. The fish attempt to dart sideways, but the striped barrier expands to block the route. They attempt to retreat backward, but the coral wall prevents it. The final option is forward, toward open water where the lionfish's head waits. The predator accelerates in a short burst, mouth opening wide enough to engulf multiple fish in succession. There is no chaotic chase across the reef. There is no prolonged struggle. The hunt is defined by restriction. The lionfish governs not by overwhelming prey with speed, but by dictating where prey may or may not go. The broader ecological twist reveals that this method of control influences reef architecture indirectly. By preying heavily on herbivorous juveniles, lionfish can reduce grazing pressure on algae. Increased algal growth competes with corals for space and light, potentially reshaping reef composition. The predator that restricts fish movement within meters of its body may ultimately influence the spatial balance of entire reef systems. Control at microscale scales upward into macro consequences. Yet lionfish are also constrained by their own strategy. Their conspicuous appearance makes them targets for human removal efforts in invaded regions. Divers spear them precisely because their slow, deliberate movements and spreading fins make them visible. What is effective against small fish becomes liability against intelligent predators. Spatial dominance does not guarantee survival against new threats. In the moment after the strike, as the damselfish numbers diminish, the lionfish's fins fold back against its body. The reef opens again, corridors restored. Other fish resume cautious foraging. But the geometry of fear lingers. Prey that witness the event adjust their distance thresholds, altering how closely they approach striped silhouettes. The lionfish's mastery lies in turning space into weapon. It does not command prey through speed or psychological illusion. It restricts the map. It blocks exits. It transforms open reef into narrowing funnel. The damselfish did not lose because they were slow. They lost because every possible path was preemptively closed. In spreading its fins, the lionfish dictates the architecture of movement. And within that architecture, prey find themselves navigating not freedom, but a carefully constructed corridor whose end has already been chosen. The reef does not erupt in violence. It goes silent. A crab steps across the coral lattice, antennae tasting the current for the chemical signatures of algae and decay, unaware that the rock beneath it has already decided the outcome. What looks like stone softens. What looks like texture becomes intent. 8 arms unfold not with speed, but with inevitability. In less than a heartbeat, every joint of the crab's body is pinned. Not pierced, not torn, pinned. The struggle does not escalate into chaos. It collapses into stillness. This is not a chase. It is not even a fight. It is a physical takeover executed by an organism whose nervous system is distributed through its own limbs, allowing it to think with its grip and decide with its touch. The crab does not die first. It loses control first. The architect of this silence is the common octopus, an animal whose dominance is not defined by brute force, but by total motor override. Each of its 8 arms contains a dense network of neurons capable of independent processing, meaning the animal does not simply send commands from a central brain, it delegates decisions outward. When an arm wraps around prey, it is not waiting for instructions. It is sensing texture, tension, resistance, and adjusting grip strength in real time. Hundreds of suckers, each lined with chemoreceptors and mechanoreceptors, create a feedback system that maps the prey's body more accurately than sight ever could. The octopus does not grab blindly. It models its captive through touch, constructing a living blueprint of joints, weak points, and escape angles within fractions of a second. The takeover begins with contact that feels like water pressure. The prey rarely recognizes the threshold moment. The octopus uses its hydrostatic musculature, arms composed of densely packed muscle fibers arranged in longitudinal, transverse, and oblique layers, allowing them to bend, stiffen, elongate, or compress without bones. This muscular hydrostat can become rope, net, clamp, or cage. When the arms wrap, they do not merely restrain. They redistribute force across multiple contact points, neutralizing leverage. A crab's pincers are powerful in isolation, but the octopus denies isolation. It immobilizes each limb individually, transforming an armed opponent into a collection of useless hinges. Control deepens through chemistry. The beak, hidden at the center of the arm crown, does not immediately crush. Instead, a small, precise bite introduces venom containing cephalotoxins that interfere with neural transmission. Ion channels in the prey's neurons are disrupted, preventing proper signaling between nerve and muscle. Paralysis is not theatrical. It is clinical. Muscles cease to obey their own nervous system. The prey's body becomes structurally intact, but functionally disconnected. The octopus does not overpower muscle. It interrupts command. Yet the physical takeover extends beyond the victim. The octopus manipulates the environment to pre-stage control long before contact. It rearranges stones around its den entrance, creating narrow choke points that reduce escape trajectories. It collects shells not as decoration but as modular barricades. It learns the topography of its territory with cognitive mapping that rivals vertebrate spatial memory, enabling it to corner prey against predictable boundaries. In laboratory experiments and field observations, octopuses have demonstrated the ability to solve complex spatial tasks, unscrewing jars or navigating mazes, revealing a forebrain architecture adapted for problem-solving rather than simple reflex predation. Intelligence here is not abstract. It is tactical geometry. The octopus's skin participates in control. Chromatophores expand and contract under neural control, altering pigmentation within milliseconds. Beneath them lie iridophores and leucophores that reflect and scatter light, allowing near-perfect mimicry of substrate. This is not camouflage as concealment alone. It is camouflage as proximity. The octopus does not ambush from distance. It becomes the environment until the prey willingly enters reach. By the time the first sucker adheres, the takeover has already begun. And yet, the narrative seems straightforward. A clever predator restrains weaker prey. The reef's hierarchy appears intact. But the reversal begins in the octopus's own body. This animal is soft, boneless, metabolically expensive. Its intelligence demands oxygen at a rate that constrains its activity. It cannot sustain prolonged chases without risking hypoxia. It lacks protective armor. It is vulnerable to sharks, groupers, and even moray eels that can tear it apart. Its strategy of control emerges not from dominance, but from necessity. The octopus must end encounters quickly because it cannot afford escalation. Physical takeover is not an expression of cruelty. It is a metabolic survival strategy shaped by the limitations of a soft body in a hostile sea. In this light, the restraint becomes a shield. By immobilizing prey instantly, the octopus reduces noise, turbulence, and visual disturbance that could attract larger predators. The arms that pin also muffle. The suction cups dampen vibration. The venom quiets the struggle. Control here prevents exposure. The octopus survives by making the event invisible. Even more startling is the extent to which control extends internally. Unlike vertebrates with centralized motor authority, the octopus operates as a consortium of semi-autonomous limbs coordinated by a central brain. Research reveals that a significant proportion of its neurons reside in its arms, granting them local reflex arcs and decision-making capacity. This distributed cognition allows the animal to multitask, one arm probing a crevice, while another stabilizes against current, and a third maintains grip on captured prey. Control is not a single act. It is a synchronized choreography of independent processes. When hunting fish rather than crustaceans, the octopus deploys a different variation of takeover. It can envelop a fish in a sudden webbing display, extending the interbrachial membrane like a parachute that traps water and body together. The fish's ability to dart vanishes. Its lateral line senses chaos, but cannot find exit. In some species, such as the mimic octopus, postural changes replicate venomous lionfish or sea snakes, altering the behavioral responses of surrounding animals. The octopus does not simply capture, it engineers hesitation. The physical mechanics of suction itself reveal deeper control. Each sucker creates a sealed chamber by expelling water and contracting circular muscles, generating negative pressure that adheres to surfaces with surprising strength. Unlike passive adhesion, these suckers can release and reattach in rapid succession, allowing dynamic repositioning. They function like adjustable clamps with tactile intelligence. When gripping rough coral, they contour around irregularities. When gripping smooth shell, they maximize seal integrity. The octopus feels its prey's microtexture and adjusts accordingly. And then there is the beak's final precision. Unlike predators that shred, the octopus often drills. Using its radula, a rasping tongue lined with tiny teeth, it can bore through shell at specific points, delivering venom directly into the nervous center. This surgical approach conserves energy and reduces risk. The prey's defenses are bypassed, not confronted. Control here is about efficiency. But the ecological twist extends further. By preying heavily on crabs and bivalves, octopuses regulate benthic populations that would otherwise overgraze algae or dominate reef substrate. Their control of individuals scales into trophic influence. Remove the octopus and the structure of the reef shifts. Sea urchins proliferate, algae patterns change, microhabitats transform. The physical takeover of one crab echoes into community-level architecture. And still, the octopus remains transient. Its lifespan is short, often little more than a few years. After reproduction, many species enter senescence driven by hormonal cascades from the optic gland, leading to self-destructive behavior and eventual death. The master of control cannot control its own biological clock. The endocrine system that once fueled precision now orchestrates decline. This is the ultimate reversal. An animal capable of overriding other lives is itself overridden by an internal program encoded by evolution. Yet within that brief life, the octopus demonstrates a form of authority unmatched among invertebrates. It does not rely on teeth, armor, or speed. It relies on interruption. It interrupts motion, interrupts nerve signals, interrupts escape trajectories. It manipulates terrain, texture, and timing. When it strikes, the reef does not explode into spectacle. It contracts into stillness. The prey does not fight because the option is removed. The arms bind like living restraints. The suckers map resistance. The venom disconnects intention from action. And in the quiet that follows, the octopus rearranges stones at its den entrance once more, resetting the geometry of its domain, ready for the next organism that mistakes stillness for safety. A shadow lunges from the reef edge, and the small fish in its path does not flee. Instead, it swells. In less than a second, a compact body expands into a floating sphere bristling with rigid spines, transforming from a bite-sized target into a jagged, inedible mass. The predator's jaws halt mid-strike, momentum collapsing into hesitation. What was prey has just rewritten the rules of the encounter. The architect of this abrupt reversal is the porcupine fish, often grouped among puffer fishes for its extraordinary ability to inflate its body as a defensive spectacle. Its control does not lie in speed or venom deployment at the moment of attack. It lies in the instantaneous manipulation of geometry. By altering its size and shape beyond what a predator's gape can accommodate, it forces retreat. Inflation is not simple swelling. The fish rapidly gulps water into a highly elastic stomach, expanding like a biological balloon. Specialized folds within the stomach wall allow it to stretch several times its resting volume without rupturing. Muscles surrounding the esophagus contract to prevent backflow, trapping water within. As the body enlarges, embedded dermal spines pivot outward, converting the fish into a spherical barrier lined with rigid deterrence. The predator's calculation changes in real time. Many reef hunters, including groupers and reef sharks, rely on suction feeding or rapid engulfment. Their success depends on the ratio between mouth gape and prey body width. When a puffer inflates, that ratio collapses. Attempting to swallow it risks choking or internal injury from the spines. Thus, intimidation becomes mechanical constraint. The fish does not overpower its attacker. It exceeds its physical limits. Beneath this spectacle lies biochemical insurance. Many puffer species harbor tetrodotoxin within their tissues, a potent neurotoxin produced by symbiotic bacteria and accumulated through diet. Tetrodotoxin blocks voltage-gated sodium channels in nerve cells, preventing action potentials and leading to paralysis. While inflation may prevent ingestion, the toxin ensures that any predator persistent enough to attempt consumption faces severe consequences. Intimidation is paired with lethality. The ecological twist emerges when considering that inflation carries cost. When distended, the puffer's swimming ability deteriorates dramatically. Hydrodynamic efficiency plummets as the streamlined body becomes a sphere, increasing drag. If a predator persists despite the display, the inflated fish is less capable of agile escape. Thus, intimidation must succeed quickly. The fish stakes its survival on the predator's risk assessment. Even before inflation, puffers exhibit bold behavior unusual for small reef fish. Their slow, deliberate swimming reflects confidence in their defensive arsenal. Unlike schooling fish that rely on collective vigilance, puffers often move independently across coral surfaces, using beak-like teeth to crush hard-shelled invertebrates. Their feeding strategy targets sea urchins, mollusks, and crustaceans, species armored against most predators. Strong jaw muscles and fused dental plates allow them to crack shells with precision. This dietary specialization influences reef structure. By preying on sea urchins, puffers indirectly affect algal grazing pressure. In areas where urchin populations are high, coral can suffer from overgrazing or bioerosion. Puffer predation helps regulate these populations, contributing to reef balance. The fish that inflates to deter predators also sculpts the invertebrate community. Inflation behavior extends beyond immediate predator encounters. Puffers can partially inflate during social interactions, signaling dominance or territorial defense. The display increases perceived body size, discouraging rivals from escalating conflict. Control through intimidation thus operates within species as well as between predator and prey. The nervous system governing this response is finely tuned. Sensory cues from lateral line detection of approaching movement or sudden water displacement trigger rapid muscular contractions. Timing is critical. Inflate too early, and the predator may adjust strategy. Inflate too late, and the jaws close before transformation completes. The fish balances vigilance with restraint, conserving energy until the threshold of threat is crossed. Another reversal lies in vulnerability during reproduction. Puffers often gather in shallow waters to spawn, releasing eggs into currents. In these aggregations, inflated displays are less common as the focus shifts to gamete dispersal. Eggs and larvae drift without the defensive advantages of inflation or toxin accumulation. The formidable adult once again depends on probability and dispersal to ensure lineage survival. The toxin itself introduces complexity in the food web. Some predators have evolved partial resistance to tetrodotoxin, allowing them to consume puffers without fatal consequences. This evolutionary arms race illustrates that intimidation is never absolute. Control is contextual, shaped by the physiological boundaries of both parties. As the reef shadow withdraws from the inflated sphere, the puffer slowly expels water through its gills and mouth, deflating back into a streamlined form. The spines settle, the stomach contracts, and the fish resumes its unhurried patrol across coral terrain. The transformation leaves no trace in the water column, only the memory of aborted predation. In shallow lagoons and reef slopes, where ambush predators lurk among branching corals, the puffer navigates with a paradoxical mixture of vulnerability and confidence. It cannot outrun many hunters. It cannot match them in stealth. Instead, it wields psychological and mechanical deterrence in a single fluid motion. The predator's retreat is not failure of hunger, but acknowledgement of constraint. A meal that cannot fit is not a meal worth risking internal damage. By altering its shape in seconds, the puffer dictates the terms of engagement. Control through intimidation is immediate and visible. It does not require hidden toxins delivered silently like those of a jellyfish, nor environmental mimicry like that of a stonefish. It is theatrical, almost defiant. The reef witnesses a small fish become a barricade. Yet beneath the spectacle lies evolutionary calculation. Elastic tissues, reinforced spines, symbiotic toxins, and neural reflexes honed over millions of years. The puffer's strategy demonstrates that dominance need not be predatory. Sometimes it is enough to make oneself impossible to consume. In a world where size often determines hierarchy, the puffer rewrites the equation in an instant. The hunter lunges expecting prey. It confronts a sphere of thorns instead. And in that hesitation, territory, time, and survival return to the small fish that refuse to flee. The strike does not happen in open water. It happens at the threshold. A small reef fish hovers too close to a crevice, drawn by drifting fragments of algae, when a jaw erupts from darkness with surgical precision. Teeth hook flesh in a flash of white, but the real capture begins after the bite. The fish thrashes, tail beating frantically against the coral face, yet instead of dragging prey outward, the predator reverses. It pulls backward. The crevice swallows both bodies. In an instant, the ocean becomes rock and space becomes suffocation. The fight is not decided by speed. It is decided by location. The architect of this spatial takeover is the giant moray eel, a predator that does not dominate by chasing prey across the reef, but by dragging them into architecture designed for their defeat. The moray's power lies not only in its bite, but in its battlefield selection. Its elongated muscular body coils within narrow reef tunnels, converting natural crevices into execution chambers. Once prey are drawn inside, escape routes collapse into stone. Moray eels possess a second set of jaws known as pharyngeal jaws, located deep within the throat. Unlike most fish, whose pharyngeal structures assist only in processing food, morays use theirs offensively. After the initial bite secures prey, these inner jaws lunge forward, grasping and pulling the victim deeper into the mouth. This mechanism compensates for the eel's inability to generate strong suction feeding in confined spaces. Instead of relying on water pressure to draw prey inward, the moray uses mechanical grip. The prey is not simply bitten, it is reeled in. The battlefield control begins long before contact. Morays position themselves within complex reef matrices where visibility is low and maneuverability is limited. Small fish navigating these labyrinths depend on quick darting motions and tight turning radii to evade predators. But when the attack originates from within the wall itself, reaction time shrinks dramatically. The eel's body remains hidden, only its head protruding slightly, mouth opening and closing rhythmically in what appears to be a perpetual snarl. This gaping behavior is not aggression, but respiration, allowing water to flow across gills in the absence of operculum pumping. Yet to prey species, the exposed teeth signal hazard, shaping distance decisions. The initial bite is powered by strong jaw muscles, capable of clamping down with significant force relative to body size. Teeth are recurved and needle-sharp, designed not to slice but to anchor. Once embedded, the eel does not release. It twists its body, leveraging its serpentine musculature against the reef structure. The confined space amplifies its strength. The prey's thrashing becomes self-defeating, as each attempt to pull free wedges it further into the crevice. Here lies the essence of battlefield control. The moray does not overpower prey in neutral terrain. It selects terrain where the prey's primary defense, agility, is neutralized. In open water, small fish can pivot and accelerate rapidly, exploiting hydrodynamic advantage. In a narrow tunnel, turning radius is restricted, and acceleration is limited by collision risk. The eel's elongated form, evolved for navigating tight spaces, becomes an advantage. What appears cumbersome in open water is perfect within stone corridors. The ecological twist emerges when considering that morays often engage in cooperative hunting with other reef predators, including groupers. Observations in certain Indo-Pacific reefs reveal coordinated efforts where a grouper signals a moray by head shaking, leading the eel toward prey hiding within coral. The grouper patrols open water, while the moray invades crevices. Together they flush prey from one domain into another. The eel's battlefield extends beyond solitary confinement. It integrates into a larger predatory network. Control becomes shared architecture. Morays also possess heightened olfactory senses. Their tubular anterior nostrils sample chemical cues carried by currents, allowing them to detect injured or stressed fish concealed within coral. Chemical detection informs spatial positioning. The eel does not wait randomly. It selects ambush sites based on likely prey traffic and scent gradients. Despite their fearsome appearance, morays lack venom in most species and are vulnerable to larger predators, including sharks and large groupers. Their reliance on crevices reflects defensive necessity. By embedding themselves within reef structure, they reduce exposure. The same terrain that traps prey shields the eel from attack. Battlefield control thus serves dual purpose: offense and refuge. The pharyngeal jaws represent a remarkable evolutionary adaptation. These inner jaws, derived from modified gill arches, extend forward using specialized muscles that propel them beyond the oral cavity. Once they latch onto prey tissue, they retract, dragging the victim inward. This system ensures that even slippery fish cannot escape after the initial grip. It compensates for the eel's inability to generate powerful suction in confined water volumes. As the captured fish is drawn deeper into the crevice, its thrashing subsides. Oxygen availability within the narrow chamber diminishes as water exchange slows. The eel, accustomed to low oxygen microhabitats, continues respiration through steady mouth opening, while the prey struggles in increasingly stagnant conditions. The environment itself becomes ally. The narrative reversal surfaces in the realization that morays are often misunderstood as aggressive open water hunters. In reality, they are architects of confinement. Their dominance depends entirely on terrain. Remove the reef, and their advantage evaporates. In featureless sand flats, morays are exposed and inefficient. Their strategy is inseparable from coral complexity. Coral reefs, in turn, are shaped by the presence of crevice predators. Small fish learn to evaluate holes and shadows with caution. Behavioral avoidance of certain structures influences grazing patterns and microhabitat use. The eel's control of confined spaces alters how prey distribute themselves across the reef, subtly influencing algal growth and coral health. Spatial dominance cascades outward. Even the eel's skin contributes to survival within tight quarters. Thick mucus layers protect against abrasion as it writhes through coral, and this mucus may contain antimicrobial properties that reduce infection risk from reef injuries. Its scaleless body allows flexibility without rigid constraint. When the struggle ends and the eel withdraws slightly from the crevice, the reef appears unchanged. A dark opening remains, quiet and still. Other fish resume cautious swimming, yet a subtle recalibration has occurred. The crevice is no longer neutral terrain. It is contested ground. The moray does not chase prey across open blue water. It does not herd schools or flash mesmerizing patterns. It chooses the battlefield and forces the fight into dimensions that favor its anatomy. Once prey cross the threshold, escape vectors vanish. In that narrow tunnel of coral and stone, the eel exerts total spatial authority. It dictates where the encounter occurs and how it unfolds. The prey may struggle, but the architecture itself denies freedom. Control is not expressed through spectacle, but through confinement. The reef is a maze of passageways, and within those passageways, the moray is sovereign. It does not remove movement through speed. It removes it through structure. By dragging prey into the tight geometry of stone, it transforms a complex ecosystem into a chamber of inevitability, where every thrash only tightens the trap and every attempt at escape collides with the very walls that promised shelter. The water convulses into a living sphere. Thousands of silver bodies flash in synchronized panic, their muscles firing in bursts of anaerobic desperation, yet every escape vector collapses as if the ocean itself has closed ranks. The school does not scatter into safety. It tightens. It condenses. It rotates in a dense, shimmering cyclone barely wider than a small boat. Around this spinning core, sleek grey forms move with deliberate calm, not striking yet, not feeding yet, simply adjusting angles with surgical precision. The fish are swimming with all their strength, but they are no longer choosing their direction. Their path has already been written. The architects of this living trap are the bottlenose dolphin, predators whose control does not rely on brute restraint, but on the systematic rewriting of prey behavior. Dolphins rarely overpower individual fish through raw speed alone. Instead, they exploit collective survival instincts, turning defensive schooling behavior into a controllable structure. The bait ball is not a random formation. It is the predictable outcome of millions of years of evolutionary programming designed to reduce individual predation risk. By attacking the perimeter in coordinated bursts, dolphins stimulate a tightening reflex within the school, increasing density to minimize exposure. Yet higher density also reduces maneuverability. The prey's defense becomes a prison of its own making. Control begins acoustically. Dolphins emit rapid sequences of clicks, constructing three-dimensional echolocation maps of fish density, depth, and movement vectors. These acoustic pulses penetrate murky water and return with detailed information about scale orientation and body mass. The dolphins do not guess where the weak points are. They measure them. Each click provides data about the edges of the school, allowing hunters to position themselves where the fish are most likely to attempt breakout. The dolphins are not reacting to panic. They are predicting it. The pod operates as a dynamic perimeter. Individuals take up flanking positions, swimming in arcs that create invisible barriers. High-speed passes along one side of the school generate pressure gradients and turbulence, discouraging fish from dispersing. Sudden tail slaps at the water's surface create shock waves that drive the school downward. Bubbles released in tight spirals form visual and tactile obstacles, exploiting fish sensitivity to abrupt contrast and disturbance. These methods do not injure, they influence. The dolphins are not merely hunting fish, they are sculpting their movement. Fish rely on the lateral line, a sensory system that detects vibration and water displacement to maintain synchronized schooling. This same sensitivity becomes a liability when confronted with coordinated predators. Dolphins create carefully timed disturbances that trigger avoidance responses in one direction while blocking alternatives. The school compresses not because it wants to, but because every signal it receives indicates that contraction is the safest option. The dolphins are effectively hacking the prey's sensory algorithms. Once the sphere forms, the tempo shifts. Dolphins accelerate through the outer layer, stunning individuals with concussive tail strikes or snapping jaws. Yet the ball remains intact because other dolphins maintain its boundaries. This division of labor reflects advanced social coordination. Research into dolphin cognition reveals large, highly folded cerebral cortices associated with complex problem-solving and social learning. Calves observe hunting techniques and refine them through practice, embedding behavioral traditions within the pod. The bait ball is not an instinctual reflex alone, it is a cultural construct refined across generations. In some regions, dolphins use mud ring feeding, a behavior in which one individual circles a school of fish in shallow water while beating its tail against the seabed, creating a ring of sediment. The fish perceive this rising wall of mud as a solid barrier and leap into the air to escape, only to be intercepted by waiting dolphins. Here, the prey voluntarily exits the water, abandoning its primary medium of survival. The dolphins do not drag them out. They persuade them to leave. The physiological underpinnings of this control reveal deeper sophistication. Dolphins possess flexible respiratory strategies, allowing rapid alternation between explosive bursts and controlled recovery. Their muscle fibers include a high proportion of fast twitch fibers for acceleration, supported by significant myoglobin stores that buffer oxygen during dives. This endurance enables sustained perimeter enforcement, while prey exhaust their glycogen reserves in continuous escape attempts. Energy economics favor the orchestrators. The fish burn through their metabolic fuel at a rate they cannot sustain, while dolphins rotate roles to conserve stamina. Yet the narrative seems straightforward. Intelligent predators manipulating schooling prey. The reversal emerges when we consider that dolphins themselves are governed by equally powerful social structures. Within pods, alliances form, sometimes involving long-term coalitions between males that cooperate to herd females during mating seasons. Behavioral manipulation operates internally as well as externally. Social bonds influence hunting roles, and dominance hierarchies can shape access to food. Dolphins control prey movements, but they are simultaneously negotiating control within their own societies. Authority is layered. Moreover, the bait ball is not a static endpoint. It becomes a focal point for multi-species interaction. Seabirds plunge from above, sharks converge from below, and other predatory fish capitalize on the compression. The dolphin's control of the school inadvertently structures a temporary feeding arena for the broader ecosystem. Their manipulation generates opportunity for competitors and scavengers alike. What begins as targeted predation becomes a communal spectacle. The dolphins dominate the choreography, yet the performance feeds many. In certain coastal environments, dolphins cooperate with human fishers, driving fish toward nets in exchange for easier access to stunned escapees. This interspecies collaboration illustrates the fluidity of their behavioral influence. They are capable of adjusting herd dynamics not only for themselves, but in coordination with another apex predator. The line between manipulation and mutualism blurs. Control becomes shared strategy. Even prey responses reveal adaptive countermeasures. Some fish species adjust schooling depth or alter formation tightness when dolphins are detected acoustically. Evolution pushes back. Yet dolphins continuously refine their tactics, demonstrating behavioral plasticity that keeps them ahead in this evolutionary dialogue. Control here is not absolute. It is iterative. The final reversal lies in perception. Dolphins are often romanticized as playful and benign, yet their true power lies in their ability to orchestrate. They rarely seize an individual and overpower it in isolation. Instead, they create conditions in which the prey's own survival strategies guide them into confinement. The fish swim frantically, believing they are escaping, while every movement reinforces the dolphin's design. As the sphere contracts to its densest form, flashes of silver intensify, oxygen levels within the ball decline, and individual fish begin to falter. The dolphins accelerate in coordinated bursts, slicing through the mass with precision, selecting targets that require minimal effort. The hunt concludes not in chaos but in calculated efficiency. When the last fragments disperse and the water clears, the illusion of open ocean returns. Yet moments before, that same water had been shaped into a controlled arena, its inhabitants herded into a structure defined entirely by predator intent. The dolphins did not merely chase. They rewrote behavior. They determined direction, speed, and density. The prey moved, but it moved according to design. Control was never about strength. It was about persuasion executed at biological scale, where instinct becomes instrument and the ocean itself becomes a stage for precision dominance. The shrimp lunges first. It commits its body to the strike with absolute certainty, antennae angled forward, raptorial legs flexed, every neuron firing toward what appears to be a drifting worm suspended in the current. The worm twitches again, an irregular pulse that perfectly matches the microspasms of injured prey. The shrimp accelerates, closing the last few centimeters in a ballistic snap. The ocean seems still for a fraction of a heartbeat. Then the seabed opens. The water implodes inward, and the shrimp vanishes into a mouth that did not exist a moment before. The predator never moved toward the shrimp. The shrimp moved toward the predator. The mastermind of this engineered fatal decision is the frogfish, an ambush specialist that does not chase, does not pursue, and rarely even swims with urgency. Instead, it constructs a behavioral trap so precise that prey willingly cross the final distance. Its body is grotesque in form, bulbous, textured like sponge or coral, often camouflaged in mottled yellows, reds, or algae greens that perfectly match the benthic substrate. But the true instrument of control is neither its camouflage nor its cavernous mouth. It is a modified dorsal spine known as the elysium, tipped with a fleshy lure called the esker. The esker is not a passive ornament. It is animated through fine muscular control, capable of twitching, pulsing, and writhing in patterns that mimic the erratic movement of small invertebrates. In some species, the lure resembles a shrimp. In others, a small fish or worm. The frogfish adjusts the tempo and amplitude of these movements based on water current and prey behavior. It does not simply wiggle the lure at random, it calibrates it. The shrimp that lunged did not act recklessly. It acted predictably. Feeding behavior in crustaceans and small reef fish is triggered by a combination of visual cues and movement frequency. Prey organisms respond strongly to irregular, short-burst movements that suggest injury or vulnerability. The frogfish exploits this bias. By presenting a stimulus that activates the feeding reflex, it overrides caution. The shrimp is not fleeing danger, it is pursuing opportunity. The frogfish's control is therefore not physical at first. It is cognitive. It manipulates the decision-making framework of its prey, steering instinct toward catastrophe. In ecological terms, this is a form of aggressive mimicry, where a predator imitates something attractive rather than something frightening. Unlike camouflage that conceals, aggressive mimicry invites approach. The frogfish becomes the object of desire. As the prey closes distance, the second phase of the trap activates. The frogfish's mouth can expand at astonishing speed, creating a rapid negative pressure event that sucks water and prey inward. This suction feeding mechanism is among the fastest known in teleost fish. The jaw apparatus hinges forward and the buccal cavity expands dramatically, increasing volume in milliseconds. Water and prey are pulled into the mouth faster than muscular escape responses can counteract. The shrimp does not simply get bitten, it is vacuumed. Biomechanically, this is a marvel of leverage and elasticity. Ligaments around the jaw store elastic energy, releasing it in a coordinated snap. The body itself remains almost motionless until the strike. From a distance, the frogfish appears inert, even comical. Yet that stillness is not weakness, it is restraint. By minimizing movement, it reduces hydrodynamic cues that might alert prey. Energy conservation is critical for an animal that relies on sporadic but high-yield feeding events. The frogfish may remain in the same spot for extended periods, waiting for the perfect alignment of curiosity and hunger in its victims. The ecological twist emerges when considering that the frogfish does not need to pursue large territories. It exerts control over a micro-environment. By occupying coral heads or sponge clusters, it becomes a focal point of risk embedded within otherwise safe feeding grounds. Reef fish often memorize safe patches where predators are absent. The frogfish disrupts this map. It inserts lethal opportunity into zones of comfort. Moreover, the frogfish's camouflage is not static. Specialized skin appendages called dermal spinules mimic the texture of surrounding organisms. Some species even host symbiotic algae on their skin, enhancing the illusion of plant life. This integration into the reef community allows it to remain within prey traffic lanes unnoticed. The lure extends outward from what appears to be harmless substrate. The decision trap operates from within familiarity. And yet, the reversal complicates the narrative of dominance. The frogfish is not a powerful swimmer. Its pectoral fins are adapted for walking along the substrate rather than sustained swimming. It is vulnerable in open water, lacking the endurance of pelagic predators. Its grotesque shape is poorly suited for flight. The decision trap evolved precisely because pursuit would fail. What appears as laziness is strategic compensation. The frogfish's reliance on prey initiative also introduces risk. If prey populations decline or become wary of lures, feeding opportunities diminish. Unlike active hunters that can seek alternative grounds quickly, the frogfish's success depends on remaining undetected long enough to entice victims. Control is effective only as long as ignorance persists. Interestingly, juvenile frogfish often resemble different organisms than adults, suggesting ontogenetic shifts in mimicry strategy. As body size increases, the lure and camouflage adjust accordingly. This developmental plasticity reflects ongoing adaptation to prey types and predator pressures. The lure itself can be lost or damaged, yet it regenerates over time. This capacity for regeneration underscores the evolutionary importance of the esker. It is not expendable decoration. It is the primary interface between predator and prey psychology. When the shrimp vanished into that opening mouth, the event lasted less than a second. There was no chase, no dramatic pursuit across reef structures. The shrimp chose to approach. It interpreted the lure as food, and in doing so crossed a threshold from autonomy to inevitability. The frogfish did not force movement through fear. It did not stun with neural overload or herd through coordination. It set a choice. The prey made it. As currents continue to sweep across the reef, the frogfish resumes its stillness, the lure swaying with subtle animation. Nearby fish resume feeding, unaware that the architecture of risk has shifted by only a few centimeters. The decision trap resets. In this quiet theater of coral and sand, control is not exerted through dominance displays or territorial aggression. It is exerted through temptation. The frogfish does not command the reef to move. It convinces individual organisms to step forward. Each strike is not an act of pursuit, but the culmination of a carefully engineered behavioral cascade, where hunger overrides caution and curiosity eclipses suspicion. The shrimp's final act was voluntary. It lunged because its instincts demanded it. And in that demand, the frogfish's strategy is revealed in full. It does not chase prey into submission. It designs the circumstances under which prey surrender themselves. The first contact is silent. A juvenile fish flicks its tail through water that appears empty, and in less than a heartbeat its muscles seize, as if an electrical current has surged through its body. The water has not changed color. There was no shadow, no chase, no visible predator adjusting its aim. Yet a microscopic battery has discharged into living tissue, and paralysis spreads faster than the fish can complete a single escape stroke. The ocean did not close in around it. It simply became lethal. Suspended within that lethal transparency drifts the box jellyfish, an organism whose control does not depend on speed, strength, or pursuit, but on turning the surrounding water into an invisible minefield. The box jellyfish does not trap prey within caves or drive them into panic. It engineers a perimeter so subtle that entry itself is surrender. Its bell pulses with measured contractions, propelling it forward with a degree of directional control uncommon among jellyfish. Unlike many gelatinous drifters that surrender to currents, box jellyfish possess complex sensory structures known as rhopalia, each containing multiple eyes capable of detecting light intensity and obstacles. They can orient toward mangrove shorelines where juvenile fish congregate, selecting environments where prey density is high. Control begins not with the tentacles, but with habitat choice. From the corners of the cube-shaped bell extend long, nearly transparent tentacles that may stretch several meters in length. These filaments are armed with millions of specialized cells called cnidocytes, each housing a coiled nematocyst that operates like a microscopic harpoon. When triggered by mechanical or chemical stimuli, the nematocyst everts explosively, penetrating skin and injecting venom in less than a millisecond. The speed of discharge is among the fastest cellular processes in the animal kingdom, driven by osmotic pressure that builds within the capsule. To the fish swimming through this region, the water appears open and safe. Light passes through the tentacles with minimal distortion. There is no scent plume to warn of danger. Unlike sharks or tuna, the box jellyfish does not chase or accelerate. It simply occupies space. Any organism that crosses the perimeter of its drifting tentacles becomes part of a biochemical equation. The venom is a complex cocktail of toxins, including pore-forming proteins that disrupt cellular membranes and cardiotoxins that interfere with heart function. In small fish, envenomation induces rapid paralysis by disrupting ion channels in muscle cells, collapsing coordinated movement. Immobilized prey are then drawn upward by contraction of the tentacles toward the bell, where the jellyfish's oral arms transfer tissue to the mouth. There is no struggle in the conventional sense. The control was established the moment the fish entered the field. What appears passive is in fact spatial dominance. The box jellyfish's tentacles form an invisible curtain that defines territory in open water. Within shallow coastal habitats, particularly near mangrove roots and estuaries, these curtains intercept small fish navigating complex root systems. Juveniles seeking shelter among tangled branches may inadvertently weave through a web of lethal threads. The jellyfish does not herd prey. It occupies the corridor between refuge and feeding ground. The ecological twist complicates this image of omnipresent hazard. The box jellyfish's transparency, so effective as camouflage, is also vulnerability. Its gelatinous body contains little structural defense. Sea turtles and certain fish species can consume jellyfish without succumbing to venom, exploiting their soft tissues as food. The predator that transforms water into a trap is itself a drifting, fragile mass that must avoid currents too strong or temperatures too cold. Its life cycle reveals another layer of control. After sexual reproduction releases larvae into the water column, the organisms settle as tiny polyps on hard substrates, often beneath mangrove roots or on submerged structures. In this benthic stage, they resemble small anemones. Under appropriate environmental cues such as temperature shifts or salinity changes, these polyps metamorphose into medusa, the free-swimming form recognized as the box jellyfish. The transformation synchronizes with seasonal conditions that maximize prey availability. Control extends beyond the immediate hunt into temporal coordination with ecosystem cycles. The venom itself shapes local food webs. Fish species that survive encounters may develop avoidance behaviors, altering swimming routes and habitat use. Repeated presence of box jellyfish in certain coastal zones can redirect juvenile fish populations away from traditional nurseries. This redirection influences predator distributions at higher trophic levels, cascading through the ecosystem. A gelatinous predator with no bones and no teeth exerts pressure on entire coastal communities. The sensory sophistication of box jellyfish further refines their environmental command. Their eyes, though simple compared to vertebrates, detect light gradients and vertical structures. This ability allows them to navigate towards shaded mangrove areas during daylight and retreat from excessively bright conditions. They are not blind drifters, but responsive occupants of three-dimensional space. The water becomes a chosen arena rather than an accident of current. The narrative reversal deepens when considering human interaction. For swimmers entering tropical coastal waters, the threat is identical to that faced by fish. Unseen tentacles in clear water. Protective stinger suits and seasonal warnings are responses to an organism that does not announce itself. Yet even in these interactions, the jellyfish does not pursue. It remains suspended, indifferent. Contact is initiated by the intruder. Control lies in invisibility combined with biochemical immediacy. Despite their potency, box jellyfish are constrained by environmental parameters. Changes in ocean temperature and nutrient availability can influence bloom intensity. In years of increased coastal productivity, jellyfish populations may surge, amplifying their spatial dominance. In leaner conditions, their numbers decline, and fish reclaim corridors once hazardous. Their control is cyclical, rising and falling with climatic and ecological variables. As dusk settles over a mangrove inlet, small fish emerge from roots to feed in open water. Among them drift faint outlines that only the most attentive eye could detect. A single brush against a tentacle halts a fish mid-swim, muscles locking as venom disrupts electrical signaling. The prey never saw the boundary it crossed. There is no roar, no chase, no visible struggle. The box jellyfish governs by redefining the environment itself. Water, the universal medium of marine life, becomes partitioned into zones of survival and instant incapacitation. In this quiet dominion, control is exercised not through intimidation, but through presence. The tentacles extend like invisible lines drawn across the sea, and those who cross them pay with neuromuscular collapse. The ocean appears unchanged. Yet within it hangs a perimeter of biochemical precision, a reminder that domination need not be loud. Sometimes it is enough to let the world come to you, unaware that the boundary has already been crossed. A reef fish descends toward what appears to be a patch of lifeless rubble, lowering its guard as it crosses the boundary between open sand and coral debris. There is no shadow, no flick of a tail, no ripple of pursuit. The rock beneath it exhales. A mouth opens from the substrate itself, and in a violent inhalation the fish vanishes, drawn into darkness by a vacuum of water and bone. The seafloor did not hide a predator. The seafloor was the predator. Buried within the architecture of coral fragments lies the reef stonefish, an organism that does not chase prey or chase rivals from territory. It erases the distinction between body and landscape. Control here is not enforced through intimidation or speed. It is imposed through perfect assimilation. The stonefish's body is an anatomical study in environmental mimicry. Its skin is textured with wart-like protrusions, and irregular ridges that mirror encrusted coral and algal growth. Pigmentation varies across mottled browns, greys, and muted greens, allowing individuals to match specific microhabitats. Even the contour of the dorsal surface breaks the symmetry typical of fish, producing a silhouette indistinguishable from reef debris. This is not camouflage layered over identity. It is morphological surrender to environment. Most predators dominate territory by patrolling it. The stonefish occupies territory by becoming inseparable from it. Once settled, it remains nearly motionless, burying itself partially in sediment or nestling into crevices. Only the upward-facing eyes betray animation, scanning through a thin film of water for passing prey. Respiration is subtle, achieved through gentle pumping that does not disturb surrounding particles. The reef does not register it as foreign. The mechanism of control is spatial. Small reef fish, crustaceans, and invertebrates use rubble zones as perceived safe corridors between coral heads. These areas provide cover from open water hunters. But when a stonefish installs itself within that corridor, it converts sanctuary into ambush point. It does not force prey into a trap. It redefines what constitutes safety. The strike is powered by buccal suction. The stonefish's mouth expands rapidly, creating negative pressure that pulls prey inward along with a surge of water. This feeding strategy, common among ambush predators, is particularly effective here because the prey's escape threshold has been lowered. The fish believed it was entering neutral ground. Reaction time is compromised by misplaced confidence. The dorsal fin carries another dimension of territorial command. Embedded within 13 stout spines are venom glands connected to sharp, grooved structures capable of delivering potent toxins. These spines are not used to hunt. They are defensive architecture, transforming the stonefish into a hazardous object within the reef matrix. Predators that attempt to swallow or bite the stonefish risk envenomation severe enough to induce intense pain, tissue damage, and systemic effects. In some cases, envenomation can cause cardiovascular distress. This defensive capability reinforces territory by deterring displacement. A predator that tests the stonefish once rarely repeats the experiment. Thus, the stonefish can maintain position for extended periods, sometimes weeks, without being forced to relocate. The reef, in effect, grants it a fixed throne. The ecological twist emerges when considering that immobility, often perceived as weakness, becomes a metabolic advantage. By minimizing movement, the stonefish reduces energy expenditure dramatically. In nutrient-fluctuating reef environments, conserving calories can determine survival during lean periods. The fish's low activity level is not lethargy. It is strategic austerity. Furthermore, the stonefish's presence influences prey behavior on a broader scale. Areas known to harbor stonefish become avoided zones for certain species, altering grazing patterns and microhabitat use. Herbivorous fish may shift feeding routes, affecting algal growth and coral competition dynamics. The stonefish's stillness radiates ecological consequence outward in subtle ripples. The venom itself represents biochemical complexity evolved primarily for defense. Unlike active venomous hunters that deploy toxins to immobilize prey, the stonefish relies on mechanical ingestion for capture. Its venom deters larger predators and accidental threats, including mammals that might step upon it in shallow waters. The spines remain erect, integrated seamlessly into the dorsal contour, ensuring that contact with what appears to be rock can deliver immediate consequence. Another reversal lies in vulnerability during relocation. When a stonefish does move, often under cover of darkness, or when environmental conditions shift, it becomes momentarily visible as a fish rather than substrate. In these moments, it loses the illusion of inanimacy and must rely on brief bursts of swimming to reach new cover. Its body, heavy and not optimized for speed, is ill-suited for prolonged escape. The same morphology that perfects disguise limits agility. Reproductive behavior further complicates its dominion. During spawning events, stonefish release eggs into the water column where fertilization occurs externally. These buoyant egg masses drift, exposing early life stages to pelagic predators. The master of territory cannot shield its offspring within rubble. Control at adult scale does not translate to generational security. Yet as adulthood returns, the cycle reinstates environmental integration. A juvenile that survives pelagic hazards eventually settles onto reef substrate, gradually acquiring coloration and texture that match its chosen microhabitat. Over time, the line between organism and stone blurs. The reef community responds to this presence. Cleaner organisms may occasionally approach cautiously, though such interactions are rare due to the stonefish's defensive posture. Most inhabitants simply reroute around it, as if instinctively acknowledging an anomaly in the terrain. Safe zones contract around its resting form. As a small fish glides over what appears to be inert rubble, it does so guided by a neural map shaped by prior experiences of safety. The stonefish exploits that neural expectation. There is no pursuit, no chase through branching coral. There is only a threshold miscalculated. The final image is paradoxical. An animal that dominates not through motion, but through stillness, so profound it rewrites the rules of space. Territory is not defended by perimeter patrol or vocal warning. It is defended by convincing the world that the defender is part of the world. Within coral fields and sandy shallows, where light fractures across broken limestone and drifting algae, the stonefish rests like a geological feature. Prey navigate by assumptions of safety encoded over generations. The stonefish waits for those assumptions to fail. In a realm where survival often favors speed and spectacle, this predator governs by erasing itself. The reef offers refuge, but only until refuge reveals teeth. Safe zones are illusions drawn in sand and rubble, and beneath them lies a body that has mastered invisibility as dominion. A ripple crosses the sand, and the ripple grows legs. What appears to be a banded sea snake, gliding with venomous authority, lifts its head and undulates in a deliberate arc, warning colors flashing in stark contrast against pale sediment. A predator approaching from the open water hesitates, recalculates, and veers away. Only then does the illusion dissolve. The stripes fragment, the snake splits into 8 limbs, and the animal that had radiated toxic certainty collapses back into anonymity. The decision has already been made. The attack has already been aborted. Fear has already done its work. The strategist behind this behavioral override is the mimic octopus, a cephalopod that does not rely on venom, armor, or speed to dominate encounters, but instead weaponizes perception. Unlike most octopus species that specialize in camouflage to disappear, this species performs an entirely different maneuver. It does not become invisible. It becomes something else. Through rapid neural control of chromatophores and extraordinary flexibility of its hydrostatic limbs, it reconstructs the silhouette, color pattern, and movement style of other marine animals known to be dangerous. The transformation is not cosmetic alone, it is kinetic. It imitates not just the shape of a lionfish or the banding of a sea snake, but their locomotion patterns, their angular posture, and the timing of their display. The result is not disguise, it is psychological coercion. Predators in coastal Indonesian mudflats operate under strict energy budgets. A failed strike carries cost, and misidentifying venomous prey can result in injury or death. The mimic octopus exploits this risk calculation. When threatened by predatory fish such as groupers or jacks, it elongates 2 arms in opposite directions while concealing the others, flattening its body against the substrate and pulsing alternating dark and light bands. This configuration mirrors the distinctive pattern of venomous sea snakes, whose neurotoxins can incapacitate fish rapidly by disrupting synaptic transmission. The predator's brain, wired through evolutionary experience to avoid striped, serpentine forms, interprets the visual cue as lethal threat. The octopus does not need venom. It needs recognition. This is control at the level of neural circuitry. The predator's amygdala equivalent, the region processing threat cues, activates before detailed analysis can override it. Fear responses are faster than careful inspection because survival demands rapid avoidance of high-risk stimuli. By triggering this shortcut, the mimic octopus hijacks the predator's own defensive algorithm. The fish alters course not because it has been attacked, but because it believes it might be. The octopus dictates movement without making contact. The mechanics behind this deception reveal extraordinary neurological investment. Like other octopuses, the mimic possesses a distributed nervous system with significant neural processing in its arms, allowing real-time adjustment of posture and texture. However, the precision of mimicry suggests not just flexibility, but learned pattern recognition. Observations indicate that individuals tailor their impersonations based on the type of predator present. When confronted by damselfish defending territory, the mimic may resemble a banded sea snake, exploiting the fact that sea snakes prey on damselfish. When threatened by different predators, it may adopt the flattened, spined appearance of a lionfish, whose venomous dorsal spines deter most attackers. This situational selection implies cognitive assessment. The octopus is not randomly cycling through forms. It is choosing the most effective psychological lever. Even its habitat enhances the effect. Muddy, low-visibility environments reduce the resolution of visual inspection, making quick threat categorization more important than detailed scrutiny. In clear coral reefs, predators may afford closer examination. In turbid flats, hesitation can mean survival or death. The mimic octopus thrives in these margins, where perception is already uncertain. It inserts itself into the ambiguity and tips the decision. Yet the reversal emerges when we recognize that the mimic octopus is, physically, fragile. It lacks the speed of pelagic hunters and the potent toxins of its impersonated models. Its body is soft, easily injured, and metabolically constrained by oxygen availability in shallow, warm waters. Mimicry is not flamboyant artistry. It is compensation. The apparent flamboyance of becoming multiple species masks a fundamental vulnerability. Its greatest strength is rooted in weakness. Moreover, mimicry does not operate in isolation. The species exists within a complex web of signal reliability, known in evolutionary biology as Bayesian mimicry, where a harmless species imitates a harmful one. For this strategy to function, the model species must genuinely be dangerous, and predators must maintain learned avoidance. If predators begin testing every striped figure and discover many are harmless mimics, the system collapses. Thus, the mimic octopus depends indirectly on the continued lethality of sea snakes and lionfish populations. Its control is parasitic upon genuine toxicity. Fear must remain credible. There are moments when the octopus abandons impersonation and chooses concealment instead, flattening against sand and blending seamlessly into substrate. This flexibility reveals that psychological control is one tool among several. It selects intimidation when exposure is inevitable, and invisibility when uncertainty favors stealth. Such decision-making underscores cognitive plasticity. In one documented behavior, the mimic extends two arms upward while the remaining limbs burrow into sand, creating the illusion of twin sea snakes emerging from a shared burrow. The predator perceives multiple threats rather than one. This multiplication effect amplifies deterrence. The octopus manipulates not just identity, but number. It fabricates a scenario in which attack appears overwhelmingly disadvantageous. The ecological implications ripple outward. By deterring predation through deception rather than violence, the mimic octopus reduces energy expenditure associated with constant flight. This conservation influences feeding frequency, growth rate, and reproductive timing. In ecosystems where predation pressure shapes population dynamics, a species capable of frequent non-lethal avoidance alters the balance of prey survival. Control through fear reshapes mortality patterns. Yet the ultimate narrative twist lies in the reciprocity of perception. While the octopus manipulates predator decisions, it too must remain hyper-aware of its audience. Its survival hinges on accurate identification of threat species. A misjudged impersonation could invite, rather than repel attack. If a predator unfamiliar with sea snakes encounters the mimic in striped form, the signal may fail. Thus, the octopus continuously interprets cues, evaluating body shape, swimming style, and approach trajectory to determine which persona to adopt. Psychological control demands psychological sensitivity. Even reproduction intersects with deception. Like other cephalopods, the mimic octopus experiences a relatively short lifespan, investing heavily in reproduction before senescence. The energy saved through successful deterrence translates into reproductive opportunity. Fear becomes a reproductive strategy. As the sun shifts and the tide recedes, the mudflat appears quiet, but beneath that calm lies an ongoing exchange of signals and counter-signals. A predator approaches, calculating opportunity. The sand stirs, and once again, danger seems to materialize from nowhere. The predator turns away before confirming its suspicion. The octopus retracts its performance, slipping back into anonymity. There is no clash of teeth, no dramatic pursuit. The encounter ends in absence rather than impact. The mimic octopus has not overpowered its adversary. It has overwritten its intention. By stepping into the predator's mental landscape and occupying the space of something feared, it governs behavior at a distance. Control is achieved without contact, dominance asserted without violence. In a world where survival often depends on force, this soft-bodied cephalopod commands through illusion, directing movement, shaping decisions, and proving that the most powerful weapon in the ocean may not be venom or muscle, but the architecture of fear itself.